The endoplasmic reticulum contains a dynamic tubular domain which continuously interacts with the cytoskeleton, undergoes constant motion and rearrangement. How the ER generates and maintains this organization is not yet fully understood. Here we present a convenient method to form a bottom-up structural organelle model for the ER, which consists of a solid-supported protein-free membrane system that can be transferred to complex lipid nanotube networks.
Hydrolysis of organic compounds for energy, protein purification, and protein extraction are not required. The only essential components are the solid substrate and phospholipids, providing the most basic ER model.
Transfer the dissolved lipid solutions into a 10 milliliter inverted pear-shaped flask, adding up to a total amount of 3,000 micrograms of lipids in 300 microliters of chloroform. Then, connect the flask to a rotary evaporator and position it with a tilt of 45 degrees. Rotate the flask at 24 RPM inside a water bath at 23 degrees Celsius for 6 hours with reduced air pressure to slowly and completely remove the chloroform. Start reducing the pressure right after initiating the rotation by steps of 20 kilopascals every 2 minutes until the pressure reaches 20 kilopascals. After 6 hours, stop the rotation and increase the air pressure again gradually by steps of 20 kilopascals every 2 minutes until reaching 100 kilopascals. Remove the flask from the rotary evaporator and add 3 milliliters of PBS and 30 microliters of glycerol. Gently swirl the flask to dissolve the glycerol.